Hello, I'm Todd Widhelm. I'm the collection manager of fungi at the Field Museum in Chicago. And today I'm going to talk to you about the biogeography of Pseudocyphellaria glabra. And glabra has this distribution, this disjunct distribution, where it's separated by the Pacific Ocean and the Tasman Sea. You can find it in Tasmania, on the mainland of Australia, all throughout New Zealand, and southern South America, and even on subantarctic islands, which aren't shown in this figure. And this distribution invokes the vicariance versus dispersal questions. That so was this thing floating away where the population's isolated as the continents drifted apart, or is this thing able to disperse across the oceans? When we look at a previous study that I did using target capture data, we estimated divergence times throughout the Lobarioideae, the subfamily in Peltigeraceae. And when we look at just the Pseudocyphellaria part of the tree and the dating on here, we see that glabra evolved within the past 5 million years, which is well after the breakup of Gondwana, which was around 160 to 100 million years ago. So by the time glabra was around, the continents are already in the same positions they are right now today. So one thing that could be driving so long distance dispersal is probably what's going on here, just based off of looking at when things evolved and using divergence times estimates. So one vector could be the west wind drift, which is a dominant wind pattern. This is a figure showing the path of a weather balloon that circled 8 times over 102 days and finally crashing down in South America. It's not hard to imagine spores also getting carried by the wind from one continent to the other. Also, migratory birds such as petrels, they all have overlapping ranges with Pseudocyphellaria glabra. Same with the albatross here, with this density map showing that that they're more commonly down in the Southern Hemisphere. Perhaps spores or asexual propagules are being carried by these birds. So I have 2 hypotheses. One is that large bodies of water, such as the Tasman Sea and the Pacific Ocean, are going to sufficiently isolate populations that they'll be genetically distinct. However, with strong wind patterns and migratory birds present down in the Southern Hemisphere, we should also see evidence for long distance dispersal. So collecting was conducted in Tasmania and in the state of Victoria in Australia, and in the beach forests of New Zealand, in the North and South Islands, and in the Valdivian rainforests of Chile. And when you just give you an idea of what glabra looks like, this is pictures of it dry, big foliose lichen. Here's it more hydrated. There's a large one right next to my head. These can be very big lichens, and they're pretty straightforward to identify. They have usnic acid, which gives them this lime green color, these brick red apothecia, and phyllidia on the margins. When we look at the undersides, the lower cortex has a chocolatey brown color, white pseudocyphellae. It's pretty straightforward to identify. And it has 2 modes of reproduction, sexual and asexual reproduction. And in order to study this, we used restriction site associated DNA sequencing, which is a reduced representation form of genome sequencing. So instead of, if this green bar is our whole genome, we're only sequencing things that are associated with these restriction enzyme cut sites, which are marked as these yellow bars. So the cut sites will then eliminate most of the genome, but we'll still get tens of thousands of loci, these rad loci, to get genetic data. And what we're looking for is single nucleotide polymorphism data, as like a SNP. So with these 3 individuals at the SNP, they're different. Everywhere else they're the same. This information is useful for inferring population structure. And since we were working with metagenomes, our rad loci were actually a mix of fungal, algal, bacterial, whatever was in that lichen when we extracted the DNA. So we had to do a mapping step, and we actually have a mycobiont genome of glabra that we had to map the reads to. And so about 20 to 25% of the reads would be fungal, the rad loci, and then we would only use those, bring those back in to generate our data sets in PyRad. And so the first thing we did was use the unlinked SNP file, which takes 1 SNP from every rad locus, puts them into an alignment, and we just used maximum likelihood to estimate a tree with it to see, will these landmass populations hold up? And they do somewhat. We see a New Zealand clade here with the green is representing the north and the tan color, the south. We have this yellow Chilean clade, this red and blue Tasmania and mainland Australia clade. But we also have this other clades over here that also contain them. So we do see structure based on what continent they're from, but it's not perfect. When we look at a minimum spanning network, we kind of get the same thing. We get some geographic clustering, but not perfect. When we look at a principal components analysis, though, we start to see a clearer picture here. So we have our Australian, Tasmania, and Victoria populations mostly falling out in this cluster over here. Our New Zealand samples, north and south, both overlapping here, and Chile kind of forming its own. Although there are still some samples, outliers from Australia and Chile kind of falling into the New Zealand area. But so this does seem to show that the first hypothesis is held up, that these large barriers, the Tasman Sea and the Pacific Ocean, are large enough to have genetically distinct populations on each side of them. And that the Bass Strait that separates Tasmania from mainland Australia and the Cook Strait that separates New Zealand, the 2 islands of New Zealand, are not large enough as we have overlapping populations. So hypothesis 1 supported. Another way to look at and rather than assuming that the genetic populations will form based on landmass, we can let an algorithm identify how many clusters are in the data and then assign the individuals to those clusters. So when we do that, we see that with this BIC score, the lower that we get, the better. And so 4 seem to have a really good fit with our data set, that it identified 4 genetic clusters. And when we look at these, we can see that group 1 is only found in Chile. It's this purple color here. Group 4 is only found in Australia. And groups 2 and 3 are primarily found in New Zealand. However, we can see individuals that are in Chile and in Australia. So it looks like we have this long distance dispersal out of New Zealand, which supports our second hypothesis that with strong wind patterns or migratory birds, we should see evidence of long distance dispersal. And with this analysis, we see that. Now, which dispersal vectors are driving the pattern? If it's the west wind drift that's primarily doing it, we would expect to see a west to east pattern. So we would expect to see Australian samples from in New Zealand, and we'd expect to see Chilean samples in Australia, but we didn't really find that. We found just only out of New Zealand. So maybe it's migratory birds because they can disperse independent of the wind direction. And another thing, it could be these floating vegetation rafts could also be playing a role. Right now, all of them could be, and these dispersal scenarios would need to be modeled in more detail to really get an idea of what's going on. So for now, it's still an open question. But moving forward though, since we have the rad loci that we sequence also contain the symbionts, Symbiochlorus. If the genetic structure is similar to the mycobiont, we would expect that to be because of dispersal through phylidia, through their asexual reproduction, because both partners would disperse together. But if we see a different genetic structure, then we might be seeing that glabra is dispersing by sexual reproduction. And also we could look at the cyanobacterial symbiont of glabra, which occur in cephalodia on the undersurface. We might expect to see an ecological effect on genetic diversity there. These are in the works. Right now we're waiting on a Symbiochlorus culture to grow up, and so large enough so we can sequence it. So stay tuned for that in the future. And with that, I'd like to thank all of you, and hope everybody's doing well. Please feel free to contact me at my email address here, and I'm happy to take questions. Thank you.